All right, this quick little video is going to give you an overview of what oxidative phosphorylation is. At this point in time, we're assuming we've gone through glycolysis, we've gone through pyruvate oxidation, and if you remember, that's also known as the prep reaction. Okay, we've gone through the Krebs cycle, okay, the tricarboxylic acid cycle, the citric acid cycle, what have you. And so at this point in time, we've generated some NADs, right? So we have two NADHs. We have another two NADHs. This is all per one glucose molecule. Okay, and so one glucose molecule feeds into glycolysis. That gives us some pyruvate plus some NADH. The pyruvate then goes into the mitochondria. It is then oxidized by pyruvate dehydrogenase. That yield is another two NADHs, right? One per pyruvate. And then that pyruvate is prepared to go into the Krebs cycle. We provide the acetyl-CoA, okay? And then that gets basically cycled into the Krebs cycle, and that really releases the remaining two carbon dioxides, right? The other carbon dioxide came out here. So, right, if you're doing your carbon accounting, right, there's 6 carbons here, there's 2 carbons here, and there's a total of 4 carbons here. So 2, 4, 6, right? So 2, 4, 1 for each of the acetyl-CoAs. That's 4 total, plus the other 2 from the pyruvate oxidation, for a total of 6, right? So there's a carbon accounting. All right, the Krebs cycle has a bunch of products, right? And one of the products is the NADH, right? There's 4 NAD, I'm sorry, I do that all the time. There are a total of 6 NADHs, okay, and 2 FADHs, FADH2s really, okay? So these, you know, there's another ATP that's generated here. There's ATPs here, talking about the straight level phosphorylation of ATPs. We're focusing on here the okay, so that's where we're starting. Start with 10 NADHs. That's what we're going to take to this next step here. So we got 10 NADHs. We have 2 FADH2s. All right, so first of all, what are these things? Remember that these are dinucleotides, right? And so if you remember nucleotides, we have our our 5-carbon ribose, and then we have some, I'm just doodling this right now for being quick, we have some, you know, some nitrogenous base like adenine or nicotinamide or flavin or adenosine, doesn't matter. Okay, and we have 2 of these, right? So there are 2 of these pairs, and just like we build DNA, right? So there's a phosphate here, and then there's the other sugar here, 5 there, and then we have our nitrogenous base over here. So this is nicotinamide, and this is just a doodle of, that's adenine, right? So this is nicotinamide adenosine, and because there's 2 of these, there's a dinucleotide, okay? Dinucleotide, and then we reduce it, we add some energy to it, and so now we're at NADH, okay? Flavin, adenosine dinucleotide, similar to that. Okay, so the job of these things is to basically act like some really strong, thick canister that can hold a tremendous amount of energy stably, and so it can actually move it in the cell, it can transport it in the cell. So what's it doing? Well, if this is the mitochondria, right, and here are the cristae, okay, the NADHs are floating around in here, the NADHs are floating around in here, NADH, there's some FADH2s, okay, and incidentally, we're not going to talk about how this happens, but the NADH that's out in the cytoplasm from the glycolysis, that finds its way in here as well, okay? And either, when it finds its way in here, it's either found as NADH or it gets converted to FADH2, right? And this is actually the more common way. This is only found in specific cell types. All right, so we're inside the matrix, and we have these very high energy molecules. So what are they doing? Well, let's say we zoom in over here. Okay, let's say we zoom in over here. So that's what we're going to do. We're going to, so here's some cristae, all right, and here's the outer membrane of the mitochondria, right? So this is the cytoplasm, this is the matrix, okay? And here's some NADH, and here's some FADH2. All right, so embedded in this membrane here, remember this is a phospholipid bilayer, embedded in here, there are several enzyme complexes, protein complexes with organic molecules. These are called, you can just think of these called cytochromes. Okay, the same kind of thing we find in photosynthesis. And basically what this complex does is it's a pump. It's a proton pump. Here's a proton, a hydrogen ion. And what this does is it pumps protons across that inner membrane. Okay, and how is it powered? By the NADHs and by the FADH2s. Okay, and so that's what the role of these things are to donate electrons to this, which is basically the electron transport chain. Okay, and so as the electron falls downhill, it's doing work by pumping the protons into this inner lumen, right? This is the mitochondrial lumen, the space in between these two membranes. Okay, so now somewhere else on the membrane, there is this amazing machine called ATP synthase. This is a huge enzyme complex. Okay, and what this machine does is it allows protons to flow downhill down their electrochemical gradient, and as this proton flows downhill, it provides a force. So this is also called the proton motive force. And what this does is it allows the ATP synthase to rotate, to spin mechanically. And what's it doing is it's taking some adenosine dinucleotide phosphate, some ADP, plus some inorganic phosphate, and it's basically latching these two things together to produce some ATP. Okay? So that's this right here, that's what then makes the world go round. That's what makes our cells go live to see another day. Okay, so the question now is, how much of this ATP can you make per, say, one glucose molecule? Well, the thing is we can't tell you exactly how many are formed, but there's a good rule of thumb. If you take this FADH2 and this NADH, for every one NADH molecule, okay, for every one of these, the amount of protons that pumps through and then in turn generates some ATP is about 2.5 ATP's worth of work. Okay, so the conversion, if you want to think of it that way, is for every one NADH, you get 2.5 ATP, more or less. Okay, and now FADH2, because it's not as high of an energy carrier, right? So it's the electron carrier is not as high in energy as the NADH is, so it's going to donate its electrons a little bit downstream. Okay, if you think of this analogy like a staircase, if NAD is donating the energy up here, and so it can bounce down and get more energy out of it, FADH2, okay, if this is NADH, FADH2 is donating it one step down, and so you're not getting as much energy coming out of it. Okay, and so the conversion factor for one FADH2 to donate its energy to go downhill here to pump in more protons that then make some ATP, you get about, for one FADH2, you get enough energy pumped across to form about 1.5 ATP. Okay? And so when you do all the math here, for a single glucose molecule, you can just say you get about 30 ATPs. Okay, now the one thing I did not talk about here is why we need oxygen. Remember, all of this is only in the presence of oxygen. So what's oxygen doing? Well, again, if this is the inner membrane of the mitochondria, okay, and these are the members of the electron transport chain, the one extra C there, okay, if this is NADH and it donates its electrons, and the electrons end up going downhill, they find themselves getting pulled through this redox, this glorified redox reaction basically. They find themselves getting pulled by oxygen. And so oxygen with these electrons, okay, with some protons, ends up giving us water, 2 molecules of water. Okay? And so without oxygen, the electrons do not pump. Without this pumping, we do not get the proton gradient. Without the proton gradient, we do not make any ATP, right? There's no ATP. Remember, this is ATP synthase. Okay, now zooming out even more, again, if this is the mitochondria, if we are not filling this with protons, right, that's where the proton pump is going here, the electron transport chain. If that's not going on and there are no more protons flowing downhill, then if you remember pyruvate out here from glycolysis, right, you got glucose being broken down to pyruvate, 2 molecules of it. The pyruvate actually gets pumped into the matrix by using the energy generated by these protons. Right? That's why we can't tell you exactly how many ATPs are generated. Okay, because some of that energy isn't going to making ATPs, but it's also going to other things, such as pumping pyruvate across these membranes. Okay, and so only in the presence of oxygen can the electron transport chain work and therefore make ATP. Okay, so because oxygen is necessary here, this process here is called oxidative phosphorylation. Okay, and so that's it in a nutshell. The oxidative phosphorylation step, this is the big payoff. Okay, this is where we make the massive amount of ATP, right? If you think about it, starting with that 1 molecule of glucose, and when it breaks down into pyruvate, right, during glycolysis, this is glycolysis, we make 2 ATPs here. Okay, if you look at the amount of free energy in a single glucose molecule, that's 686. That is not 680. 686 kilocalories. Okay, that released from that when we measure it. Okay, the amount of energy in 2 ATPs is about 14 kilocalories. Right, so when you do the math there, that's about 2.1% of the free energy. That's terrible, right? So only in the presence of oxygen do we get that additional about 28 ATPs. Okay, so these 28 plus these 2 give us about 30 ATP. And when you do that math, that's about 30% or so efficiency. And that's about as good as we're going to get. Okay, so that's oxidative phosphorylation in a nutshell.